Hello. 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 Right, thank you for coming to tonight's True Talk. This is the first of our viral series of... Oh no,
I was saying no technically.
okay. Right, this is our first lecture of a viral series, and this is Kayla Peck from UNC. Yeah. And she's going to tell us a bit about the basics of viruses and viral evolution. And like usual, if you have any questions, just ask in the middle of the talk. We like a discussion going on the floor, so do not feel shy about raising your hand, basically. So without further ado, here's Kayla.
Okay, how's the hearing volume? Okay, cool. So thanks for the introduction. I'm Kayla. I'm a grad student in the biology department at UNC. I work with Christina Burge, Ralph Berg, and Mark Heisey, and kind of my main focus is looking at viruses and virus evolution. So because there's two upcoming virus talks, one on Zika virus and one on HIV, I'm kind of focusing on more of the basics of viruses. So what are viruses? What can they infect? Kind of just giving us a really strong foundation, so that way when you go to other talks, you can have a really good understanding of what viruses are and what you should be paying attention to when they're introducing these viruses. So to start off, when we talk about viruses, the first thing we can ask is, okay, what actually is a virus? So if you look at the formal definition of a virus, it's actually a small infectious agent that replicates or reproduces inside the cells of other organisms. So if we start off with the beginning, okay, small, how small is small? We can look at this diagram of different sizes of organisms or components of organisms. So over here we have a frog egg, it's 1 millimeter diameter, you can see that with your naked eye. After you go down, each of these ticks is a 10-fold reduction in size. And so here we can see flu virus, so it's about 10-fold smaller than the average bacteria. You can't actually see that with your naked eye or even a light microscope. You're going to require an electron microscope in order to see that. So you can see here that viruses are kind of the smallest agent that are required before you get down to like smaller things like atoms. So going back to this definition, we have small infectious agent. So here the words infectious agent are very carefully chosen, and that's because one of the biggest debates when you talk about viruses is the traditional question, is a virus alive? So if we ask this question, let's first think about what components compose a virus. So here are two diagrams of viruses. Here's a naked virus, which is just composed of nucleic acid, so the genome of that virus. Viruses can be either RNA or DNA viruses. So here's the genetic material that will tell you what that virus is going to be. Surrounding that nucleic acid will be a capsid. So the capsid is just basically a protein coat that surrounds and protects that genetic material. Over here we have an envelope viruses, so these are kind of the two structures that can be possible. This envelope virus has an extra layer around it that's made up of, it's like a lipid bilayer. This envelope will be a little bit of the cell that it emerged from, but again, that's just in order to protect the capsid and the nucleic acid, so really you only have those three things. And then sticking out from the envelope, you can have different proteins, and those are what's going to interact with the cells that it infects. So if we think about,
can the same virus have both the envelope version and the non-envelope version?
So the question is, can the same virus have a naked version and an envelope version? To my knowledge, no. A virus species or species will be either just naked or enveloped. So if we now go to like what biology says are the definition of what is life. These are the components that are required to be met in order to say whether something is living or not living. So we have cellular organization, reproduction, so can it produce more of itself? Metabolism, does it produce waste? Homeostasis, can it regulate its internal forms? So for humans, something with homeostasis would be like regulating your body temperature. Heredity, do you produce something that looks like you? Response to stimuli, is there something in the environment that you can react to? And then growth and development. So if you look at each of these components and think about, can this nucleic acid with a capsid around it or an envelope meet any of these requirements? And for a virus on its own, it can't meet any of them. And so from this definition, you can say, okay, a virus isn't alive. However, if you now add the caveat of, well, with the help of a cell, now suddenly it can reach all of these requirements in order to be considered living. So so with this, kind of, you've got to go with your own personal opinion on whether or not you think adding this caveat with the help of a cell actually allows you to say that a virus can achieve all these requirements. So personally, I think a virus can be considered living because there's a lot of organisms that are obligate symbionts, so two organisms that require each other in order to actually survive. And so if you make the exception for those organisms and say, okay, well, with the help of their obligate symbiote, they can achieve these, then I think you can make the exception for viruses. But aside from that argument, kind of my definition as an evolutionary biologist is something alive is something that evolves by natural selection. So viruses do evolve by natural selection. And the reason why you add this kind of by natural selection on here is if you just said something is living if it evolves, then you kind of get into this weird realm of computer programs that have been shown to evolve. But they don't evolve by natural selection. So by having this definition, which I think evolutionary biologists are getting more attuned to, it allows viruses to be life. Yeah, Chris?
So, can you say though, if you were to put me in the middle of the ocean and all the way down, that I wouldn't be able to satisfy some of those things as I could die? So wouldn't you say that someone, a particular organism or something that's alive would have to be in a particular region for it to accomplish those things anyway?
Sure. So the question was, if Chris is floating in the ocean, he's going to die and not be able to reproduce or have anything that's hereditary, etc. So yeah, that's true. You do require nutrients or something from the environment in order to achieve all of these things. But the thing is even with nutrients or in any kind of environment outside of a cell, a virus can't accomplish these. So the general consensus of is a virus alive is the answer, there is no real consensus. And so if we go to this really popular virology blog, it's hosted by Vincent Racaniello up at Columbia. He runs this really popular podcast called TWIV, or This Week in Virology. And so he posed to his audience whether or not they thought viruses were alive. And this is kind of what that broke into. We got yes, no, something in between, and I don't know. And so kind of the answer is it's based on what you think. I mean, I think when this argument comes up as a virologist, you're kind of like, well, it doesn't really matter. Like saying whether it's life or not life is just, a human classification system. And so you can study viruses and know everything without actually having to say whether or not it's alive or not. Okay, so moving on from whether or not a virus is alive, I've told you what the components of a virus. I just wanted to take some time to compare a virus and a bacteria, since I think generally they do get kind of mixed up and convoluted a lot. So if we go over some of the differences, a virus is not a cell and a bacteria is a cell. So as I mentioned before, a virus is just the genomic material and a protein capsid that surrounds it. Whereas bacteria actually has a cell wall, it has structure within that cell. And that cell wall is a bit more rigid and stable than a virus particle would be. A virus can't survive on its own, and a bacteria can. So even though if it was in a vacuum without resources, as Chris kind of mentioned earlier, neither of them would survive. If you have just like a blanket of nutrients, virus will just sit there and do nothing, whereas bacteria can reproduce and survive. The thing that draws them together, which I think is the source of a lot of the confusion, is the fact that they can both make you sick. So let's say you go into the doctor's office and you have a really, really bad strep throat. You can either have mono or sore throat. Strep throat. So mono is caused by a virus called Epstein-Barr virus, and strep throat is caused by bacteria called Streptococcus pyogenes. So you go in and they say, okay, well, which one is it going to be? And hopefully, if you go in these days, they'll take a swab or maybe draw some blood and try and figure out which one of these it is. And the reason why is because you'll have the same symptoms based regardless of what's infecting you, but the treatment is going to be extremely different. So one of the differences between viruses and bacteria is the fact that bacteria can be treated with antibiotics and viruses can't. So the way antibiotics work is by preventing the cell wall of a bacteria from synthesizing. And since viruses don't have a cell wall because they're not a cell, that kind of treatment does not work for them. And in fact, with something like mono and strep, treating it with antibiotics can actually make your mono infection worse. So I think if it has a symptom of like a really severe rash, I don't necessarily know the mechanism, but if you go in and they give you antibiotics and you get a really bad rash, you probably actually have mono. And for mono, there's no antivirals, and so you basically just have to wait it out. But how many people have had mono in here? Oh man, you guys are, okay, yeah. I've heard it's really horrible, so, I mean, don't envy you. Oh, and also, like, mono is called like the kissing disease because you get it from making out, but it's okay, sir, because you can actually get it elsewhere. It's like not just saliva, you can get it from other things too, so don't be embarrassed. Okay, so we've thought about the differences between virus and bacteria. So what can viruses actually infect? Viruses can infect humans or other mammals. So here we have an example, HIV, which can infect humans. Viruses can infect non-mammals. So here we have avian species. This is influenza A virus, so H5N1, if you've heard of that, it's the avian flu. It can infect avian species. Here, viruses can infect amoeba. So this is what's known as Mimi virus, which for a while was the largest virus ever discovered. So here we have 400 nanometers. And back, if you remember that graph that I showed, influenza virus is about 100 nanometers, and so it's much larger than your normal virus. And then finally, viruses can infect bacteria. So this is what's called a bacteriophage, and you can see this is kind of your characteristic, really charismatic form of a virus where it looks like a little, I don't know, robot or spaceship. So it will actually go down and kind of land onto a bacterial cell with this landing pad, and it will shoot its DNA down into that bacteria. So it's very creepy, but very cool, I guess. So here we can see that there's a huge diversity of species that viruses can infect, and then there's also a large diversity of what those viruses actually look like. So when we think about what viruses can infect, the next thing we can ask is, okay, where do you actually find these viruses? So obviously you can find them in organisms, so they can infect a wide variety of cell types. Here we have lungs, they can infect neurons, they can infect skin cells, red blood cells. You have viruses that maybe can infect almost every organ type. You also have viruses that can be present in vectors. So some viruses will infect you just by you kind of inhaling a particle, but some viruses actually require an insect in order to transfer that virus to a new person. So these are viruses that will be in your blood. So a mosquito will come over to you, you have the virus, it'll suck your blood, get the virus along with the blood that it sucks up, fly off to someone new, suck their blood, and give you the virus. So there are a small number of species, mosquitoes, sandflies, ticks, that are able to transmit viruses. You can find viruses in the air and the surface. So here is a lovely photo of someone just sneezing, and there's a bunch of virus particles that kind of get ejected during that. You have viruses that you can find on surfaces. So it's estimated that influenza virus can survive on a surface, just as that, virion, for 24 hours. You can find viruses in the soil, so there's a lot of bacteria in the soil, so a lot of the viruses that you find there will be bacteriophage, viruses that infect bacteria. And then you can also find viruses in the ocean, and this is kind of one of the more expanding fields of virology, where they're finding that there's an intense amount of diversity present in the ocean, so they call them marine phages. And these viruses will not only infect bacteria, cyanobacteria in the ocean, but they'll also infect things like amoeba, algae, all sorts of species. So I think it's been estimated that there are about 10 million virions present in one drop of seawater. And so with that, viruses are considered to be the most abundant organism agent present. And for more fun numbers, they say that there's estimated about 10 to the 31 viruses on the planet, and that's like 10 million more than the number of stars in the universe. So viruses are basically the most predominant form of life or non-life, depending on your personal view. Okay, so now that we know a little bit about what viruses can infect, where you can find them, I wanted to point out that viruses can be composed of either RNA or DNA genomes. So here we have kind of DNA, our classic double helix. We have our 4 common nucleotides. RNA has a uracil instead of a thymine, and that's where the nucleotide difference arises. And here you can see this is a depiction of a single-stranded RNA. Now, if we go back to biology, where we think about the central dogma of DNA, you go from DNA to RNA to protein within an organism. And so if this DNA virus gets into your system, it can go DNA, RNA to protein. if an RNA virus gets into your body, it can go RNA to protein. So some of the differences between RNA and DNA viruses are the fact that RNA viruses bring in some of their own machinery in order to complete the replication cycle, and DNA viruses will use all of the host cell's machinery pretty much. And so this is because if you go back to this DNA, RNA to protein, that's what humans do. We're comprised of DNA, we go DNA, RNA to protein. So this virus doesn't need to do anything extra. It can just come in, hijack everything that we have, and use it to complete its cycle. RNA viruses, however, even though we go RNA to protein, what these guys need to do is also go RNA to RNA in order for them to make more RNA to package inside the virions that then get produced. And we actually don't have that because we don't need it for anything. So RNA viruses have to bring in something that will allow it to go RNA to RNA, to replicate RNA, to then package into virions that will go in the rest of the cells. In addition to that, RNA viruses typically have small genomes. DNA viruses have larger genomes, and this is mostly based on the fact that RNA is a little less stable than DNA. And that's kind of one of the hypotheses about why humans or all organisms are DNA, and I think one of the largest reasons behind that is because DNA is a lot more stable than RNA. And then finally, RNA viruses have higher mutation rates than DNA viruses. And this is because if we go back to that thing where I was saying that our viruses have to bring in to go RNA to RNA, that can't, it doesn't have proofreading capabilities. So while your RNA virus is making another copy of itself, it can't say, oh, I've made an error, I need to go back and fix that. And so you have a lot more errors, whereas DNA is using the DNA machinery that we have, and so it says, okay, there's an error, I'm going to go back and fix that, just like our genomes would. So if we think about the differences between RNA and DNA viruses, here are some of the examples of which of these viruses fall into either RNA or DNA. So hopefully you guys will recognize some of these up here. We've got Epstein-Barr virus, which I just mentioned, causes mono, herpes, HIV, influenza, norovirus, which is your classic cruise ship virus, causes puke. Yeah.
So the mistakes that are made with the RNA virus, like the flu virus, is that's the reason why every year we're going to get a new flu vaccine where they say like it's a different strain and all this stuff, because it's constantly making mistakes and it's essentially a different virus.
Exactly. So the question was, is the reason why RNA viruses have high mutation rates, is that why influenza you have to get a new vaccine every year because it's a new strain? And that's absolutely correct. The influenza viruses that are circulating are mutating really fast, and so in the span of a year, they've mutated and changed enough such that the strain that you see a year later isn't similar enough to the strain in the previous year, and that's why the vaccine isn't working. Yeah.
Is it easier or more difficult to make a vaccine against one versus the other?
So the question is it easier or more difficult to make a vaccine over one or the other? I'm not aware of any formal studies that have looked at that, but I think it has more to do with how the immune response reacts to those viruses rather than whether or not it's RNA or DNA. So when you can get a vaccine for a virus, it's because you're able to infect it with something similar and your body elicits an immune response that then protects you in the future.
Is there a difference though in the immune response between one versus the other?
As far as I know, I'm not sure of anything that is specific between RNA and DNA viruses. I think what's more important is what the surface of the virus looks like. So whether or not that capsid is there or the envelope and kind of what's sticking out from that. Okay, so looking at these viruses, another component that's important to think about is we have what their genome composition is, but then we can also think about what kind of infection they cause. So the two options are acute or chronic. And so acute will be something that's rapid or short-term, something that makes you sick but then is cleared rather quickly. And so if we think about this where our y-axis is the amount of virus in an organism and x is the time, you can see that it'll be, okay, you get sick, but then your body clears it, and that's your end of the infection acute. Chronic is more of slow, long-term. And so here, if we're looking at the graph, you get sick and then you don't actually clear that virus, or it takes a really long time to clear the virus. So if we look at these viruses on the side, here would be our acute viruses. So influenza, you get sick and then you're better in a week or so. Norovirus, well, rabies virus, that's a whole other story. Zika virus, you recover and that virus doesn't stay with you forever. Slower long-term viruses, chronic, we have HIV, herpes. So if anybody has ever had a cold sore that's caused by herpes, you'll have kind of recurrent episodes of those cold sores because that virus never leaves you. So it just gets kind of reactivated, and that's why you'll get a cold sore.
Ebola is supposed to be staying for a while.
So he said Ebola is supposed to be with you for a while, and it's like 9 months. Okay, I'm not quite sure about the time frame, but 9 months in the grand scheme of things like HIV, which is with you your whole life, I guess they have classified it as acute. Okay, so when we talk about everything I've just covered and someone presents a new virus, so okay, you're going to hear this talk on Zika virus, these are the things that they're going to tell you, and these are the things that are kind of going to give you a framework by which to consider that virus and how it causes disease. So the first thing will be what type of genome is it, RNA, DNA. The second will be what's the size of that genome, so how many genes are potentially acting in order to produce and cause an infection. What kind of infection is it, acute or chronic? What cells does it infect? That'll let you know where in the body it's infecting. And then what are the symptoms? So how can you tell if you're actually infected with this virus? And so we're going to do a small case study looking at these things. So we're going to look at varicella zoster virus. Who's here been infected with that? Thanks guys. Okay, so we got some virologists in the back there. What virus is this? Chickenpox. All right, now who's been infected with varicella zoster virus? Awesome. Okay. So we look at the virus that causes chickenpox. It's a double-stranded DNA virus. Has about 125 kb genome, which is reasonably large for a virus, but small in the grand scheme of the world. So humans have a genome size of 3 million kilobases. What kind of infection? It's acute. You get your chickenpox, and then within a small time frame, they're gone. It infects lung cells, and then it eventually migrates and gets into your epithelial cells, which is why you get kind of the skin rash. And again, you have the pox-like symptoms of chickenpox. And so, for those of you who have been infected with chickenpox, I'm sure you know that you can't get chickenpox again. And so what's kind of the rationale behind why you can't get chickenpox twice? If anybody wants to shout it out.
You build up immunity?
Yeah, okay, exactly. So you said you build up immunity. So once you're infected with varicella zoster virus, you get chickenpox, your body recognizes it and says, okay, I'm going to build these immune system components such that if I ever see this virus again, I'm going to be able to get rid of it. And so you can't get that infection twice. This is also the premise behind why vaccines work. Okay, but chickenpox is actually a really unique virus in the sense that you actually have a chronic stage of this virus where it infects your neurons. So does anybody know what this is? Virologists aside. No hands. Yeah, exactly. Okay, so we got shingles called out here. So chickenpox, it has this acute phase, so it'll get in, give you the rash, but then it actually goes into your nerve cells and it stays there forever. And so if you've been infected with, or if you've had chickenpox, you now have this virus in your nerve cells. And so it can be activated pretty much like at any point. No one's really certain what actually activates shingles. There's people that think maybe stress, name your hypothesis. So shingles can arise. I mean, it's pretty dangerous for elderly people. Has anybody actually had shingles? Okay, so I actually had shingles like 3 years ago, and the reason I went into the doctor is because I had this rash on my leg, and I thought I was having an allergic reaction to a bug bite, and I'm like, oh my gosh, I'm allergic. They're gonna have to amputate my leg because there's just these shooting pains that go up all your nerves. And so I went in, and then when they told me it was shingles, I did not tell them that I studied virology naturally. So yeah. So anyways, that's kind of my public service announcement. Okay, so thinking about why you can't get chickenpox twice. Now there's a vaccine for chickenpox, and I believe there's a vaccine for shingles. Yeah. Okay. Great. So
does that mean if you have shingles, you have to infect other people with chickenpox?
So the question is, if you have shingles, can you infect other people with chickenpox? And the answer is yes. If you have shingles, you're not supposed to go around children that have not been infected with chickenpox. I mean, there is a little, the way it's transmitted, it's not super efficient. So if you have shingles and you have a small sore somewhere and you go around a child, it's not instantly going to get chickenpox, but yes, you can give them the virus. Okay, so thinking about this idea of when you encounter a virus, you now have immunity, so you can't get infected by it again. This leads us into thinking about vaccines. And so the discovery of vaccines actually came from smallpox. So smallpox is a really horrible virus. It gives you small lesions which turn into scabs. It's kind of like chickenpox, but each of those bumps will turn into this weird scab, and inside that scab will hold infectious virions. It has a 30% mortality rate, and when you die, it's mostly because you have this hemorrhagic version of smallpox, which is where blood leaks from your organs. So please don't Google hemorrhagic smallpox, I beg you. Okay, so the reason why vaccination was even invented or discovered, because you imagine, trying to talk to someone, okay, we're going to cure you of this by infecting you with it, and obviously that sounds unintuitive and psychotic. But Edward Jenner noticed that milkmaids actually got smallpox less frequently than the rest of the community. And so he started thinking about, okay, this is a very weird phenomenon, what's the explanation? And the explanation is that once he started looking at the udders of these cows, he found that they had cowpox. So these are kind of small lesions on the udder of the cow. And when the milkmaids would go out and milk the cows with cowpox, they would get small lesions on their hands. And so they were getting infected with cowpox. The cowpox wasn't lethal. And so with that, they were getting enough of an infection that their body was preparing their immune system such that if they encountered smallpox, they would now be protected against it. And so that was kind of the foundation of how he was able to discover vaccination. And this was in the late 1700s. So smallpox actually is eradicated. And so the last round of smallpox vaccines, I believe, was administered in 1972. So not to make things awkward, but does anybody have a smallpox vaccination in this room? Yeah, okay, so if you have a smallpox vaccination, because it has a small lesion or a scab, almost everyone who has that vaccination will have a little circle scar on their shoulder. So I've got a prop here. If you want to go see the scar, just ask him later. But you can also ask, most of your parents will have this smallpox scar. So it's really interesting to think about. This is something that our generation has never thought about, but all of our parents will have this very similar scar. Okay, so about vaccinations, half of you in this room will know that I cannot talk about that without gushing a little bit about Louis Pasteur, who's pretty much the best scientist ever. So Louis Pasteur, although he's known for pasteurization, he also discovered the rabies vaccine. So rabies is the most lethal virus in the world, untreated. So without treatment, rabies has a 100% fatality rate. And I did add this little tilde because I'm sure the virologists back here will call me out and say, I think to date maybe there's 6 people that underwent this really weird treatment after having rabies and they survived, but it's 100%. So if we compare that to other deadly viruses that we think of, we have the hemorrhagic version of smallpox. It's about 95% mortality rate. HIV is 80%. Ebola is 70%. And again, this is all untreated. So Louis Pasteur, there's this really bad rabies virus. The symptoms of being infected with rabies is basically the most horrendous thing you can ever imagine. So it infects your nerve cells, so it'll go up into your brain. You kind of lose all sense of who you are. You have a really strong case of hydrophobia, and so you can't drink water. You'll have a cup of water and you'll be dying of thirst, and your hand will be shaking, and you literally cannot force yourself to drink water. It's the weirdest thing. And so anyways, there's a lot of other symptoms, but we're a little too graphic to go into at this moment, so if you want to talk about it later. Also, I forgot about this, but I have some books up here that if you guys are interested in some of the topics that I've talked about. This is Rabid. It's basically kind of the historical and cultural history of rabies. It's really interesting, so you can come up. There's also one about Louis Pasteur up here, just so you know. If you have the itching for some more knowledge. So in his attempt to create a vaccine for rabies, he basically gathered the saliva from rabid dogs, and then he passaged that virus in rabbits multiple times. And the reason he did that is because if you think back to kind of this cowpox, smallpox protection, you can't infect someone with rabies and then have them be protected against rabies because it's such a horrible, deadly virus. What you need is that virus to be changed just slightly enough that you'll get an immune response to rabies without actually getting rabies. And so I think in vaccine culture, this is called, you either need to be attenuated, so a weak virus, or inactivated, it no longer causes illness. So by passaging this rabies virus within rabbits, he's attenuating it for human infection. And then he infected a boy named Joseph Meister in 1885 who had rabies and was 100% going to die. So we tested the vaccine in him, and it worked, and now we don't have to worry about rabies ever. Yay! Okay, so now...
vaccines can work after you've been infected.
That's what yeah.
Oh, great. So she said vaccines can work after you've been infected. So some viruses, rabies is the best example I can think of in my head. If you get the vaccine before the virus actually has a chance to kind of have like a larger titer inside of you, so actually infect and get within your neurons, then it can protect you. So if you get bit by a rabid dog and you go in the next day and get the rabies vaccine, it'll protect you from the rabies that was in the dog's saliva that's now going to course through your body. If you wait like, I don't know, I'll say a week just because I don't want anybody in here to like get rabies, then you're essentially, it won't work. But that's not true for every virus.
How did he attenuate that and then inject it into rabbits? What did he do?
Oh, so he basically just got the saliva from the rabid dog. It's actually, I'm glad you asked, because he would have these like leather gloves while he's going out to wrestle these rabid dogs, and he had his like lab assistant there with a pistol, and he's like, all right, if I get bit, just shoot me. And you're just like, okay, like I'm not that dedicated to science, like maybe I shouldn't be doing this. But yeah, so he would get the saliva, then he would inject it into a rabbit, and then the way he would collect it from the rabbit, he would actually get it from their umbilical, or their spinal cord, sorry, umbilical, spinal cord, and extract it and then infect the next rabbit with it.
Oh, okay.
Yeah, so going through multiple rabbits in succession is what allowed it to be attenuated.
Which is where rapid succession came from.
Oh, glob. All right, so next thing to talk about before we get into the virus evolution component is biosafety levels. So for working with any infectious agent, bacteria or viruses, you basically have 4 levels, with 1 being the safest and 4 being the least safe. So BSL-1 will have things like, BSL for biosafety level, will have things like E. coli, non-pathogenic bacteria. BSL-2 will be things that are influenza A virus, measles, hepatitis, and this is what you would wear, like a lab coat, gloves, or glasses, that's about it. BSL-3 is a little bit more dangerous, so this will be things like SARS coronavirus, West Nile, the avian flu, H5N1, and you'll be required to wear a mask. So these will be things like that might aerosolize easily, so you would like inhale them, or things that don't have any drugs or vaccines present. So SARS coronavirus is one of those that fits both of those categories. BSL-4 will be things like Ebola and smallpox. So since upcoming is a Zika virus talk, also it's just been in the news a lot, what BSL level do you guys think Zika is?
3, right?
2.
Okay, we got some 3s, we got a 2. So it's actually 2. And the reason is because even though it doesn't have a vaccine currently, for most individuals, if you got infected with it, you kind of just have malaise and it wouldn't be that bad. The whole microcephaly thing aside, which we can talk about later. But also, Zika is transmitted by mosquitoes. So in order to get infected with that, you would literally have to like inject with a needle into your bloodstream. So it doesn't aerosolize, and so that's why you can work with it just kind of at a normal level. What about HIV?
2.
3.
Yeah, no, great. So, good job, Haley. So HIV is also 2, and that's for a similar reason. 1, there are treatments for HIV. I mean, there's no cure, but you can get treated for an HIV infection. But also, since it's transmitted via blood, you know, you just don't use needles in the lab, and then you're fine. Kind of thing. Yeah.
Could a tick not transmit Zika?
I'm sorry?
Could a tick not transmit Zika? Do we know?
As far as I know, a tick would not be able to transmit Zika. Really, there's only 1 species of mosquito that's capable of infecting or transmitting Zika.
I hear now there might be 2.
You're probably more up to date than I am. She said there might be 2, so we'll say less than 35. Yeah. Okay, so and then the virus that I work with, and actually Anne and Jake back there also work with dangerous viruses, so maybe stay away from them. Just kidding. I work with MERS coronavirus, which is a BSL-3 pathogen, and so I'll talk a little bit more about that at the end if we have time. Okay, so now that you guys have kind of your primer of what a virus is, we'll start to talk about virus evolution. So I have here that it's scary but neat. And evolution for viruses, the scary component is that they evolve extremely rapidly, and this has a detriment on human health. So you can imagine kind of the example that was brought up earlier. You have the flu virus, it changes every year, so you have to get a new vaccine every year. And because they're changing so fast, and because vaccinations take a little bit, take a long time to produce and distribute, they're actually predicting what that vaccine is going to look like before the virus actually hits. And so that's why if you get the flu vaccine and it doesn't protect you, it's because, well, I guess they guessed wrong that year. So the scary part is that it has this negative impact on human health. The neat component is that viruses will evolve on in a time frame that we can actually witness and document. And so we can start to ask and understand what kind of evolutionary processes will shape populations as they evolve. And so this is an example from Trevor Bedford at the Fred Hutch Cancer Center in Seattle. So it's nextflu.org. I recommend everyone check it out because you can actually go and interactively filter like current data on the flu. So this is a phylogeny, so showing relationships between flu viruses, and you can basically go and look at, this is last year's flu. So here are the different strains, different components of flu, what it's actually looking like to an evolutionary biologist.
What does the process mean?
Oh, I think the process where maybe these are the lineages where they tried to vaccinate against. I'm not quite sure. So if we ask the question of how did viruses actually evolve in the first place, so there are these really unique agents. Again, they're not quite living, not quite non-living. If we think about how they evolved, normally when we talk about organisms and how they evolved, we look at this called the phylogenetic tree, so the same version that I had there, just a little bit different. So we can use sequence data combined with some fossil data to say, okay, humans are more closely related to cats than they are to these other species. Avian species are more closely related to reptiles than they are to these other species. And sharks or fish are kind of out here, not related to anything closely. And so this is how we're able to say what evolved and kind of what this evolutionary history looked like that led us to the current species that we see. But why doesn't this same approach really work for viruses, if anybody wants to guess?
You don't have a fossil?
Exactly, you don't have a fossil record, so that's one. Okay, wait, I'm gonna die first. So he's correct. He said that the mutation rate is so high, and that's 100% correct. So with viruses, you get this thing called genome saturation when you try and construct phylogenetic trees. And if we look at an example of that, so let's say we have a human genome and a virus genome, and these colors, let's just say, represent nucleotides. As we go along in time, our viruses have high mutation rates, so they're going to be changing rapidly. So here we have a change, here we have a change, here we have a change. Okay, humans finally got a mutation, viruses are still changing. Okay, so we now look at it kind of on this later time frame. If we look at the number of changes compared to our original genome, they both say 1. However, here we have one change that occurred, and here we have a whole slew of changes that occurred. So when you have genome saturation, you can't really tell what happened between here and here, but that's all the information that you have. And so because of this, we can't construct these kinds of relationships because the viruses were just mutating so fast, we don't know what was happening in the middle, and we can't tell what they're most closely related to. So there's actually 3 different hypotheses based on, or that say how they think viruses actually exist, or how they originated. And if you're interested in talking about those afterwards, I'd be happy to go over them. There's no real consensus on which one is the most correct. It's one of those things, again, where everyone has their opinion, and there's not a lot of data saying that one is more correct than the others.
Kind of 2 questions. One, well then, how do you genetically define one virus? This is what one virus is genomically, and then at one point, do you get so many mutations that it's no longer that virus?
So that's a great question. So the first one was, so if you have a lot of mutations, what defines a virus? And the answer is that, or virus species, and the answer is that there's no real strict rule. I think some people will go on the basis that, because if you think about in the biological world, they'll say, okay, a species is one that can reproduce with another one, and that's really simple. For viruses, it'll be based on genetic identity, so they'll say something like, okay, when this virus is 90% different from other viruses in that family, now we can call it a new species. But it's kind of, there's no real rule, it's a little hand-wavy. In terms of when do you get so many mutations that it turns into a different virus, I mean, even though viruses are mutating really fastly, a lot of these mutations are going to be deleterious. So there will be some selection to kind of keep the same structure of a virus. How many mutations will turn it into a different virus depends on the genome size, depends on what those mutations are. I really have no idea. Great question though. Okay, so what can viruses evolve to do? The first, most obvious one is that they can evade the immune system. So here's kind of a virus particle, and this is an antibody. It's binding to that virus particle, and now that virus particle can't get into a cell. If that virus happens to get a mutation that now, let's say, adds this large chunk of carbohydrate onto its surface, now the antibody can no longer bind there, now it can infect your cell. So this is how viruses can evolve to evade your immune system and make you sick. Viruses can evolve to become more or less virulent. So if you have something that's more virulent, you're going to be more sick, you're going to stay in bed. And so that's not really good for something like influenza, which is transmitted by aerosols, where you need to be out and like hanging out with your friends and like feeling like okay, but still like coughing all over them. And so that's when a virus would evolve to be less virulent. So you're just, you're out and you're up, you're not incapacitated. For these viruses, this is more like if you required a vector to transmit the virus. So you can be like laying there not feeling great, but a mosquito can still bite you and transmit the virus. So an example of evolution that's happened kind of recent or within our time frame is that in the 1950s, Australia was kind of overrun with this rabbit population, and so they decided to infect the rabbit population with this myxoma virus, which was lethal to rabbits. So you infect it, it'll die. They kind of infected a bunch of rabbits and like let them loose, and then when they came back, what they found was that the virus had evolved to be less virulent. So you can imagine from the virus's perspective, it's not necessarily beneficial to immediately kill the host that you are living in. Like you want it to at least transmit before you kill it. And so the virus became less virulent, like less deadly, such that they now came back to a lot of viruses, or a lot of rabbits and a lot of viruses that don't kill those rabbits. So cautionary tale for when you try and use biology to, control biology. Quote me on that. So finally, viruses can evolve to infect a new species. So here we have perhaps an influenza virus, starts out as an avian virus and then can evolve to infect all of these different species, which influenza can infect today. And this is what I'm going to focus on for the last bit of the talk. So my research really focuses on looking at how and maybe why viruses are able to start in one species and then expand to infect a new species. So since viruses do require a host in order to survive and replicate, then just by intuition, they can expand to infect a new species that they previously hadn't been able to infect. So just again, bringing back influenza, influenza started out as an avian species, and then by different methods, they were able to jump into humans. So here we kind of have a pig as the intermediate host. Pigs are often called like mixing vessels because you can get a lot of different influenza viruses infecting them. And then, you could remember the swine flu outbreak of 2009. So in addition to these anthropocentric examples that we also have documented cases of other host range expansion events kind of within our time frame. So parvovirus actually originated as a feline virus, and then I believe in the 60s was able to adapt into dogs, which is what we typically associate the virus with today. So coronaviruses, which I mentioned earlier, is the virus that I work on. And so here we can have just some different pictures of coronaviruses. Coronaviruses are called that because I guess when the first person looked at this image, they thought it looked like it was having a crown, and corona is crown in Spanish. So if we go through that table that I presented earlier about what people are probably going to tell you when they're introducing a new virus, it's a positive sense single-stranded RNA virus. It's 28 to 32 kilobases, which again is very small in the grand scheme of the world, but when we did the chickenpox virus, which was DNA, that was about 125 kb, so it's small in the world of viruses. Another layer, it's actually large for an RNA virus. So the second largest RNA virus is 20 kb. So coronaviruses are the largest RNA virus. It causes an acute infection, so you'll probably get a cold for most coronaviruses, and then it'll clear. It infects your lung cells, and again, you'll get a cough or a fever. Actually, most human coronaviruses don't get diagnosed as such because it just kind of looks like a cold or a flu infection. So if we look at coronaviruses, they actually have expanded their host range to infect new species multiple times over the course of their evolutionary history. So these are 4 human coronaviruses that have emerged to infect humans. They've jumped into the human population between 100 and 800 years ago. And most of the originating species was bat, although we do have an example where it was a bovine species here. So these viruses started out infecting bats and then somehow were able to gain the ability to infect humans. And these 4, if you haven't heard of them, is because they cause mild symptoms. Like you've probably been infected by these and not really known it because they just don't get diagnosed that often. If you've heard of coronaviruses, the ones you probably have heard of are the ones that cause severe disease. So the first of these is SARS, severe acute respiratory syndrome coronavirus. It originated in a bat and then in 2003 emerged into humans. The exact path that this virus took in order to get into humans is not well known. However, it's thought that a civet intermediate host played a role. So these are these small cat-like creatures that are in China, and they thought that perhaps the civet was exposed to bats kind of out in the rural areas, and then those civets are brought into the Chinese marketplace where, you know, they're used for food, and so you get a lot of fluids that are then transmitting the virus to people. SARS infected over 8,000 people with a 9% mortality rate before it was able to be controlled by public health measures. So more recently we have MERS, or Middle East Respiratory Syndrome coronavirus, which also originated in bats, and then in 2012 emerged into humans. Similar to SARS, while SARS had the civet as that potential intermediate host species, here it's camels. So the first person to be infected by MERS was a camel jockey, and so they began looking at camels. They were able to find antibodies in the camel's immune system that suggested that they were able to be infected by MERS. But again, we don't really know exactly how or why this virus was able to get from bats into humans. And so if we think about this landscape, we have MERS, this new virus that emerged really recently. We know it can infect bats. It can infect humans. And then kind of the next step that the Barrick lab does is to say, okay, can you study this like in vivo? How is this virus causing disease and what can we do about it? And unfortunately, MERS does not infect mice. And so mice are the traditional small animal model. Without them infecting mice, you can't really study how this virus actually causes disease. So when it was found that mice couldn't be infected, it was then found that other small animal models couldn't be infected. So we had our ferret, our guinea pig, our hamster. So none of these animals can be infected by MERS. So we're kind of like at an impasse, and how do we actually start to study this virus and begin to develop drugs or therapeutics. So just to note, MERS is currently still circulating in Saudi Arabia. It has a 36% mortality rate.
Can it infect monkeys?
It can infect monkeys, but the difference there is that it's really hard to work with monkeys. It takes a lot of time. I think the marmoset is currently kind of the best.
I was just thinking like, yeah, monkeys are, mice are like the equivalent like medicinal model that people use. And not because they're hard, but...
One monkey is 25 grams. Sorry.
Yeah.
At least says one monkey is 25 grams, so... That's a lot of paper. Okay.
What about small animals that aren't rodents?
I'm not sure anything is... So she said, what about small animals that aren't rodents or in this family up here? I don't think anybody or anything else is really used or is tractable to use. So in order to use one of these animals, you have to have a breeding facility. You have to have, things.
Just a human.
I'm sorry? No. Provided this guy.
No, people use cats.
People use cats. Okay.
And there's a whole lot of other animal models that are amenable.
Sure.
We have them here at UNC.
Yeah, so she said people use cats and dogs and other animal models. That's definitely true, and you could use that maybe in the short term to figure things out. But the main goal is to get something smaller that's more tractable to work with, that multiple labs and not just the ones that are equipped to work with it.
Stuff.
Oh yeah.
Is there any viruses that are targeting specific kind of racial components, or they're particularly susceptible to, it's called Middle Eastern?
Sure. So she asked, are there specific viruses that may be more or infect a certain demographic more than others? So this virus emerged in the Middle East, so are the Middle Eastern people more susceptible to this virus? That's a great question. It's something that people are looking into, but I'm not sure if there is an answer to that. That is something that I've thought about a lot, because you see MERS-like coronaviruses in South America, in Mexico, in South Africa, but you don't see anybody getting infected there. So there's something weird going on. My project going in with this landscape was, how do we, why can't MERS infect this mouse, and what can we do about it? And so when we think about why can't MERS infect a mouse, we can think about what actually blocks infection. So here's a very basic life cycle of a virus. And when I say basic, I mean there's a lot of complicated components here that you really, in each of these steps, there could be something going wrong in the mouse. And so how do you actually start to understand which of these matters? So if you think about the first thing in this entire life cycle, it's really looking at the virus getting into that cell. So if we look at this zoomed up, we have a virus that's interacting with proteins on the cell, it gets into that cell, and then it releases its genome here and then makes more of itself and does all the downstream stuff. So if we look at this component here, receptor binding, entry to the cell, release of the genome. So if we think, okay, the first step that could go wrong is receptor binding, so we're going to look at that. So for MERS, here's an example of the crystal structure of the mouse version of the receptor and the human version. So human in blue, mouse in red. The receptor for MERS is this molecule called dipeptidylpeptidase 4, DPP4. Here we have it modeled looking at the portion of the virus that's actually going to interact with the host cell receptor and utilize it in order to enter and infect. So based on some protein modeling, we were able to identify two differences between the human and the mouse molecule that we thought might be important for explaining why MERS can utilize human DPP4 but not mouse. So to test that, we went into an experimental system. So this is representing the human version of DPP4, and we express that molecule in cells and then take them into the BSL-3 and then infect with a virus that's tagged with a red fluorescent protein. So when you see red, that's an indication of infection. So you can see here we have our human version being expressed, we see a lot of infection. Our mouse version being expressed, we don't see infection. Now when we take those two changes that we identified and introduce them into the backbone of the mouse molecule, we now see high levels of infection. Hopefully you guys can see the red back there. So we took a molecule that couldn't be utilized by MERS, and now we've made a molecule that can be utilized by MERS. And this combined with other experiments suggested or helped to provide evidence that it actually is a receptor level issue and not something else within the animal. Yeah.
What was the nature of the amino acid changes?
So the question was, what is the nature of the amino acid changes? One of the changes stabilized an important hydrophobic core within the receptor binding domain, so the virus component. They found that if you disrupt the hydrophobic core in the virus, you don't see infection. The other one, it actually removes a glycosylation site. So if this large carbohydrate glycan on the surface, when we knocked it out, suddenly now the virus can get down and kind of bind there. Thanks for asking, because that was a lot of work. So from this, we were able to say two residues on their own are capable of mediating infection of the mouse receptor by MERS. And so with this information, there's a slew of things that happened after. The first is that we created a mouse using CRISPR-Cas, so this genetic editing system, such that we could have a transgenic mouse that was capable of being infected by MERS. Okay. My projects that kind of followed up on this were looking at why those two mutations mattered, and also what if that same mechanism applies to those other species that could not be infected. And I'm happy to chat about that after. Okay, so to summarize kind of the work that I've done looking at virus evolution, looking at host range expansion, we have this landscape of a newly emerged pathogen that can infect bats and humans, but it doesn't infect any of these species. We identified two changes that were important in the receptor of the mouse that prevented, or that's when we made the two changes that allowed the mouse to be infected, and then we created a transgenic mouse that now is capable of being infected by MERS. So that's step one. If you have a virus that can infect mice, you can one, make a mouse, which seems weird, but we did it, I guess. And then two, you can make a virus. So is it possible to engineer the virus or adapt it or evolve it in order to infect mice? And so to do that, when we look at this setup where we know that there's these two changes that are important for preventing the virus from utilizing the mouse receptor, we can make the hypothesis that you'll need multiple mutations in the virus in order to allow it to actually infect the wild-type mouse receptor. And so going into an adaptation experiment, I wanted to take these mutations and basically see if I can passage the virus and select for a virus that would be able to infect a wild-type mouse. And even though we have the transgenic mouse, you can see that the value of this would be, when you're breeding mice, if you have this one lineage, you kind of have to wait until that one lineage is ready to do your infections. Whereas if you have a virus that can infect any mouse, you can now look at different mouse species, you can cater it, or lineages of mice, you can cater it more to more labs being able to actually use this to study. So we started here where we had our mutations that caused a lot of infection, and then we would slowly transition that passaging onto this version of the receptor that was unable to be infected. So it looks something like this. We start out with 100% of this, and then we slowly move to 100% of that, passaging the virus in the hopes that we would select for something that would be able to infect this.
Is this process dependent on the background strain?
On the background strain?
Of the mouse? Because they have such different immunity and everything. I mean, did you focus on a T57 Black 6 or a valve, or what did
So
you
the
use?
question is, does this experiment depend on the background of the mouse? And so for this, I didn't mention it, but this is actually done in vitro, so this is just using cell lines. The transgenic mouse that they made was, one specific background of mice, and so ideally, we would take the virus from here and make sure it was compatible with that background lineage, because that's,
Because they have such differences in their
yeah, definitely. Is there another question back? Okay. And so kind of just to go through the result, basically I passaged this 25 passages, which is basically, around 3 months, and didn't see any kind of adaptation. And so based on this negative result, we can go back and say, well, this supports our hypothesis that multiple mutations are likely needed in order to produce a virus that's capable of infecting a wild-type mouse. So if we have our mouse, can't be infected, again, we identified 2 mutations you can make in that, Vincent.
So you were saying that for influenza, for example, you can predict the roadmap, how it would evolve, right?
You're saying if the adaptation experiment had worked.
No, so for influenza, you were saying that you can predict the evolution roadmap.
Yes.
With this one, in a computer, can you predict what would be required or how much time it would take for it to evolve?
Okay, so the question is that for influenza, like I mentioned earlier, for the vaccines, you have to predict which strain is going to be most prevalent, make the vaccine for that. For an experiment like this, can you do computer models to predict how MERS would evolve or how you could get a mouse-adapted virus? And the answer is no, and that's because we have decades of influenza data out there, and influenza comes in cycles, and so you can kind of map the cycles of which strain is most prevalent and cater to that. For something like MERS, we don't have that data, we don't have cycles of infection, so it's not comparable. Does that make sense? Okay, thanks. So we identified 2 mutations that you can make in the backbone of the mouse receptor in order to allow MERS to be able to infect it, and from that we were able to make a transgenic mouse. If we want to now make a virus that can infect a wild-type mouse, we hypothesized that multiple mutations would be needed in the receptor binding domains, the part of the virus that interacts with the cell. After adaptation, we didn't see anything that achieved this requirement. And so one of the avenues I'm exploring is whether or not you can use a mutator. So have a virus that has an elevated mutation rate, because maybe that would then allow the population to produce the genetic variation required to get the multiple mutations needed. On a more realistic level, I think that what could happen is that when we did this modeling, we didn't actually have the crystal structure of the mouse version of this molecule, which adds some noise into that analysis. So if someone in a structural biology lab crystallizes the structure of the mouse version, then we could potentially use computer models to more accurately predict what virus we could engineer to infect a mouse. Okay, so that was a lot of things. Yes?
Sorry, just a quick answer. How homologous are those proteins in mouse versus human?
How similar?
Yeah.
So you said how similar are the human and mouse molecules? I think they're about 85% amino acid identity.
The structure is almost the same. Even 40% would be almost the same.
I mean, so we use a threaded structure. He said the structure is the same. We use a threaded structure, but earlier we noted that the glycosylation site was what was important to be removed. So without a crystal structure, we didn't know that there was a glycosylation site there, or glycan. And so with things like that, the glycosylation site is a huge barrier, and you can't really know how the virus can restructure around it without knowing specifically what its components is. are. Make sense? Okay, we can chat later. Okay, so sorry that was a ton of information, but if I wanted you to, walk away out of this room with just remembering anything, first, viruses are tiny and they require a cell in order to survive. If you guys want to have a debate afterwards whether or not that means life or not, I mean, that would be probably a lot of fun.
All right.
But the answer to that is that no one really knows if they're alive. I mean, or not knows, no one really agrees. Viruses,
What makes a dead virus?
So she asked what makes a dead virus. So that would be a virus where maybe the capsid has been changed enough that it actually can't interact and infect a cell, or the genome is so messed up that even if it got into a cell, it wouldn't be able to produce more of itself. So a lot of times when they say like an inactivated virus for a vaccine, it's because they've taken like a huge chunk of that virus, the genomic material, out, and so it can't actually function as like a replicating particle.
Let's say if it's flu virus, so they can survive for 24 hours on the surface. It's because they dried out after 24 hours. So
Right.
if we add water to the surface again, doesn't mean it's reactive.
So she said if you leave like I said earlier, if you have flu on the surface for 24 hours and it dies, is it because it dries out? Yes, desiccation is a huge issue for why viruses will no longer function. If you add water to it, I don't know, and I don't want to be quoted, so, I don't know. But yeah, good question. Okay, hang on. Okay, so viruses can be found anywhere. They're in the air when you are sick and sneeze. They're in the ocean, the soil, in your body, etc. They're extremely diverse. Hopefully, you guys can take that away. So we have things that look like these little, spaceship pods that will infect bacteria. We have things that infect plants that look very broad-like. We have influenza virus. There's a lot of diversity, not only in the genomes and how they function, but in how each virus looks. And so when you're studying viruses, it's really hard to draw conclusions for one virus and apply it to the others because you have this intense diversity. Viruses can expand their host range. No one's really sure why or how. And what I mean with that is we had this example of these viruses that were going from bats into humans, but what was actually the reason why they were able to get from bats into humans? Was there some specific selective pressure that drove them to make that change? Was it just by chance? No one's quite sure of what the actual history is when we see something like MERS that jumped from bats into humans. We just know that it infected one species and now it infects another one. And then finally, MERS research is cool, everyone get into it. Okay, and with that, thank you to my labs, thanks for the invitation to speak, and I will take questions. Oh yeah.
Can you eat a virus? Like, can you get it through ingestion? I gotta cook my chicken all to shit just to, prevent salmonella. Can I kill viruses? Can they be on food? Can I cook it and will they die?
Yeah, so the question is can you eat viruses? And the answer is you probably have eaten like 500 million viruses today. So yeah, there's bacteria on your food, there's viruses that infect those bacteria. There's plant viruses, they're probably in plants. So yeah, there's so many viruses, it's just that, the large majority of viruses aren't going to make you sick. So viruses are an important part of kind of like any ecosystem, if you will. So I'm sure your natural gut bacteria have bacteriophages that are also important for controlling them.
There's this sci-fi idea of a killer virus that wipes out humans. How likely do you think that is?
Oh boy. Okay, so he said, how likely do I think it is that a virus will evolve to wipe out everything? The answer is, I'm not sure. So I think that, the Ebola scare wasn't that realistic because a big component of that is that people weren't managing transmission very well. So, not being aware of touching saliva, the fluids, everything. So that doesn't bother me, doesn't worry me. I think something like the H5N1, the avian flu, if it is able to evolve or transmit or get into humans, the reason why it hasn't had an outbreak is because it can't transmit between humans fast or well. If that potentially evolved too, the only thing stopping the virus would be its ability to evolve, kind of the example with the rabbits, where it would have to evolve lower virulence as time went on, so it didn't actually wipe out its original source. If it has a backup source, like it's still infecting waterfowl and it doesn't care that humans are a reservoir of virus, I don't know. And yes, I am saying this so you will all fund virus research.
Sorry, this isn't my area, but you could talk about the ethical considerations when you're talking about adapting a virus to become promiscuous within like a wild population that is also present in human populations. So the virus that you were talking about, it infects humans, it didn't infect mice. If you make it to infect mice, the populations of humans that we're talking about are lower income, underserved resource populations from a public health standpoint. Like, what kind of containment parameters are in place to make sure that that doesn't reach wild mouse population, which would have a detrimental public health effect?
Right, so the question is about ethics in terms of adapting a virus to a new species, the mouse here. And so if we think about the concern to the human population, as you mentioned, essentially if you adapt a virus to mouse, you're adapting it away from humans. And so if you have a virus that infects mice very well, the probability is extremely high that it just won't infect humans very well. That combined with kind of the actual containment facility that's in place, the possibility of an escaped virus from this adaptation is, I mean, I don't want to get like overconfident, but I'd say it's like 0. I mean, in terms of, do I feel like I'm a monster if I make a virus that can infect a new species such that that species dies and previously wouldn't? Of course I feel like a monster, but you know, that's just the choices I guess you gotta make. Yeah. All right. Oh, yeah.
So do you expect, if you're actually able, so the transgenic mouse that you made,
yes,
and the sort of evolutionary push that you've made in your passaging, would you expect, I'm just asking, like at the end of the day, would that be a similar model to what, say, humans would get? Because you're now pushing towards a mouse model, and mice don't normally get infected. And I mean, do you get feedback from, I don't know, like reviewers and things like that that say, wait, this isn't a really good model? I'm just curious.
So I don't think so. So mouse adaptation of viruses is pretty common. So
is it? Okay.
Even things, or even viruses that naturally infect mice will still undergo some adaptations such that they're infecting mice that's that mimics how the infection would be in a human.
Okay.
And so that's kind of the phenotype that you drive towards. Yeah.
Is it just that you're changing the capsid protein that allows it to do the rest of it so it means attack, and that's why it's a better model?
Yeah, so ideally you would see changes in the spike protein, or the protein that's interacting with the host cell receptor, and then there's probably going to be changes elsewhere in the genome, but those aren't ones that are important or that you care about. So you would just take the evolved spike and introduce it back into the original backbone, such that everything else would be the same as the virus that infected humans. Yeah.
What's the sequence identity, protein sequence identity between your mouse and your human?
For the host cell receptor, it's 85%. Yeah. The structural identity is extremely similar.
Well, that's based on the molecule.
Yeah, it's based on threading of the molecule.
Right.
Yeah. So yes, very fast. All right, thanks guys.
Yeah. Another round of applause for Kayla for an awesome talk. Thank you guys for coming out. This is a great turnout. I hope I will see you guys in the next virus talk, which is going to be the last Monday of August, which is the 29th of August, I believe.
Yeah.
Come out, we'll be talking about Zika virus. It will be given by Helen Lazier from UNC Chapel Hill, who had recently made a mouse model to study Zika virus, and so she'll be giving us an update on the Zika virus and how it's going to affect all of us, which I think is going to come to this house. But anyway, before the last pint, please tip your waiters who bring food to your table if you have ordered food, and please come out for the next talk. Thank you all. Oh, one more thing. So we do have the return of the suggestion box. So there are slips of paper in there. If you have a topic that you would like to hear about, anything that is like kind of related to science. I was thinking about science and music at one point. So if you want to hear about any topics, write it down here and let me know. Or if you have any recommendations of speakers, also write it down, please. And I will try we will try our best to recruit what you want to hear. Thank you. Thanks for organizing, John.
Thank you.